Welcome to Laplink Connection. I'm talking today with Dr. George Korch, who is, and I have to read this because it's really difficult, he is the Laboratory Director of the National Biodefense Analysis and Countermeasures Center and the President of the Battelle National Biodefense Institute. And of course, he was a former acting assistant secretary of the Department for Health and Human Services, longtime director of the Army CDC, and a professor at Johns Hopkins University. I mean, if I would read more, we will spend 20 minutes. Hi, George, how are you?
Good, Thomas, how are you?
Very good. Well, today we want to talk a little bit about COVID, but not about today and all the political discussions, but rather to look beyond the current one. So when we look at the situation, we saw that 2020 was the year of COVID. 29 was H1N1 or swine flu. Very different outcomes, very different pandemics. The question is, how can we better prepare to face new pandemics in the future? What steps should government and science take to be better prepared?
Great questions. It's been the question for the ages ever since my time early in the military. Question is, how do we prepare for emerging threats or coming threats? And some of them, quite frankly, won't be natural. Some of them could actually be man-made because biotechnology is moving at such a fast pace. But the answer to your question is there has been a lot of progress between 2009 and 2020, 2021, which has prepared us and allowed us at least to be more responsive now than we even were in 2009. And the things that have sort of propelled or accelerated our pace or ability have been things like these platform technologies for vaccines. So vaccines have probably had the biggest emphasis and push on the technology side. Therapeutics lags behind. And surprisingly, diagnostics, which should probably have been at the forefront, should have been the easiest thing to do, was the thing that unexpectedly really gave us kind of the strike. Why were the diagnostics not there right away? Because those are platform technologies as well, and it doesn't take a huge amount, especially when it's a nucleic acid based or a PCR based diagnostic that everybody's now familiar with. Those things should have been easy, out there in bulk. And so the lessons learned, if one thing we learn, it's, well, what did we not do right? And what can we do better the next time around?
Yeah. I mean, clearly the vaccines were produced as a record time. Was this because science was faster, because they had more money, more focus, more urgency? Or was the time savings because of cutting regulatory red tape?
No, great questions. All of the above. On the science side, I mentioned these things called platform technologies. Think about it as a software package that has sort of a basic framework, but now you can plug in whatever you need as the situation changes. Well, it's the same with some of these vaccine platforms. The older vaccines were basically made from growing up a bunch of the pathogen and then inactivating it, or finding a variant of the pathogen that was attenuated. It didn't cause disease, so it was safe even though it was live. Those are the old style vaccines. The vaccines of the last 15 years or more recently are based on taking SNPs of the DNA or RNA of the virus or the bacteria that are responsible for the immune system to say, oh, I can protect using this piece of information, or using vectors. A vector is a completely different virus, like one of the J&J vaccine that we will be seeing in the EUA, emergency use authorization, probably in a few weeks, is based on an adenovirus. It's a completely different virus. It causes a common cold. They've changed the virus so that it can't replicate, it can't reproduce in you, but it can carry the message of the coronavirus on its surface, and your immune system sees it and says, oh, this is coronavirus, but it's not. It's a vectored vaccine. So all of those platforms have been under intense scrutiny over the last 10, 15 years. In 2014, Many of these were brought out for the first time to try to see if we could get ahead of the Ebola virus outbreak in West Africa. And a lot of them cut their eye teeth there. So 3 or 4 vaccines made it into the field in big numbers back then. Zika, people kind of probably forget about Zika, which was about 2.5, 3 years ago. The mRNA vaccines, the vaccines right now that are Moderna and the vaccine from Pfizer. Those concepts really started to accelerate because I was on these committees looking at those vaccines for Zika. But because it's a platform, you just use the backbone and you just plug in what you want. So science has advanced. The government, this time around, used all of its mechanisms to de-risk, if you will, the financial and some of the regulatory burden. Regulatory burden never goes away, ever. You always have to have safety as number one, and then efficacy is important to demonstrate, is there any value in taking this, or is it just dishwater? Even if it's safe dishwater, if it's not useful, don't produce it. But the government de-risked the costs for these vaccines. Vaccine or a medical countermeasure can take anywhere from about $900 million to up to $2.5 billion to bring to market. So by government saying we will underwrite some of the costs of production, some of the ability of you guys to get out there and do large-scale clinical trials, because that's the most expensive thing of those phase 3 trials. That was a big, and speed, the time. If we produce these things in big batches rather than do small batches and then another small batch and another small batch, but if you had a big batch made right away, if it was looking good, then you just said, full speed ahead, bottle this and we'll send it out rather than you have to go back and remanufacture. And then on the regulatory side, as I mentioned, over the course of the years, things like emergency use authorization. This is the first time in a big way that we used an EUA to put forward a vaccine into the population. EUAs were established initially for biodefense. In other words, somebody was going to do something bad to you guys or us guys, and you needed to get something out fast, and maybe it wasn't going to get licensed, but you had this use permission. Again, because you had safety and everything taken care of, it was before licensure. These vaccines still have to be licensed to be able to be used without sort of this specific veil of permission, but at least they're there. So those are 3 major areas, and there are a few others, and the last one that I'll point out is there are enough cases in the world to test these vaccines. Before, you would have a small population, or the disease would come and go so fast that you couldn't go out and do what are necessary. That the gold standard is double-blind, placebo-controlled trials. So one group gets the vaccine, the other group gets a placebo, and then you look to see how much disease occurs in each of the groups, and statistically, is there a difference? And that's what powers the ability of these vaccines to be determined to be efficacious. So those are the things that really played out in this particular pandemic.
Yeah. So, that's quite clear. And I can see the question of government, because we saw that different regions of the world approved it at different times. It was the same vaccine. All the documentation was there, but you had to do red tape there, red tape there. The EU did the approval in late December, and so they were late starting with the vaccine. So going back to science, so I think it's very clear what you're saying is that we are really getting smarter in producing vaccines with so much more advancement in science, and that probably goes also for diagnostics and therapeutics as well, right?
Yes. On the diagnostic side, there are so many different types of mechanisms now for identifying with great precision the type of pathogen that's in there. Almost everybody now can tell you what PCR is, whereas 15 years ago, they would have thought, it's a new bank down the corner. But that amplification technology or other ones that change around, whether you need to have fancy equipment or you can do it at home in a dipstick, those are the big advances that are being made. The most important thing about diagnostics, of course, aside from specificity, which means it's identifying the right thing, it's specific to the thing that you're looking for, and sensitivity, that means you can detect it in a range that's important. So if only 10 bacteria cause an infection, but your device only detected 1,000 or more, well, then you don't have the sensitivity to say this person's infected. So those are the two big parameters on diagnostics. But after that, it becomes ease of use, cost of goods, the same thing that happens obviously in software. How easy is it to use? What's the price point for it? And when you finally get these things all worked out, if we can get things as fast as possible to the end user and have the data collected and readable in a way that everybody can make bigger interpretations, and not just what does it mean for that person, but for an infectious disease, what does it mean for the whole population? Those are the important parameters on diagnostics. And then therapeutics really is the toughest of all of them, in that these viruses and bacteria and other pathogens have evolved to live on our good graces, to parasitize us. And they have many, many tools at their disposal to try to get around all the defenses that we put up, not just the immune response, but other things. There's called innate immunity, and there are other mechanisms that we use to shut down or kill cells that are infected and things like that. So some of those things are very important to our own cells. So there's toxicity that could be involved in producing a therapeutic that handles that virus really well, but it also has a negative side effect for the host. So that's the toughest of all, the drug side of the house. But there have been platform technologies that are moving forward, and I think I've mentioned to you in the past that I really think we are on the threshold with all of the fantastic informatics that we have, bioinformatics and accumulation of gigantic data sets and machine learning, and looking at deep patterns, that we may be able to find ways to intervene at the level of the pathogen in terms of its specific targets, its genome, and the host in terms of what it has, its patterns and pathways for its own biosynthesis, and where the parasite is using the host's capabilities for doing what it needs to do, and target them in a way that we have much broader capability, so they don't have to have like a silver bullet, one shot, one kill type of thing. But you can do it in combination, combining things. And the best example of that is HIV, where we have controlled disease by having 4 different drugs that act simultaneously on the pathogen, each in a little amount. So it's like a death by 1,000 cuts phenomenon. We need to do that for many more diseases.
Yeah, I think that's great insight. I'm just thinking that the world of science certainly has evolved from looking through your microscope, as Robert Koch did, and looking what he finds, versus now science is so much more medical science, but also big data yes. centers. I mean, we know that already from the weather forecast. Yeah, it's not looking at the clouds and saying, yeah, well, it will rain, but it is masses of data that goes in, and they need the supercomputers. So, yeah, and so with the platforms, I think what you are saying also is that we're really getting smarter in producing vaccine for the next COVID, because we had already different COVIDs. Swine flu was COVID, right? So we can do faster detection, faster coming up with vaccine in the future.
Yes. I'm going to borrow a phrase I once heard at a biological conference, basically, and it was somebody getting up and saying, "Look, biology or medical treatment isn't rocket science. It's much harder than rocket science." So we kind of walked away from that feeling a little proud of ourselves that the challenges really are the big data analytics. Not forgetting that it goes the other way as well. Understanding deeply the particulars about one pathogen. What do you need to know about it? Where is its weak points? So yes, those are important. And the other point that you were making, I can't remember the end of your phrase. But okay, we'll move on from there.
But I mean, you gave me another piece for perhaps my last question. In this time, how did you experience work? Were you forced to home office with your team? But I assume that labs and science installation cannot just work from home. You're not sitting alone at your laptop and working. I guess science workers are the same frontline heroes as well as many others, don't you think?
Certainly in all of the technical people, all of the manufacturing people, the scientists, technicians that were pursuing what do we need to know about this particular virus. Yes, they were definitely frontline workers. My own laboratory is responsible for assisting the FBI on cases where there are biological materials, and we need to know not only what it is, but can we tell who may have made it. And my threat characterization center, this is right up their alley. This is a brand new threat. What can you tell us about it? We did all of the work on the issue of how long does it survive in the environment. So those data points that you see about sunlight having an effect where it's knocked out, in full sunlight in a matter of minutes versus in the dark at cool temperatures, that was all of our work. We're looking at aerosols. We're trying to identify just how much virus does it take to infect you. I mean, we're wearing masks, and that's good because you're impeding the airflow. But against how many? Do you need to protect against 1 million or 10? A lot harder to protect against 10 than 1 million. So yes, my crew has been at it. In fact, boy, they were working 7 days a week for months on end. I just can't speak more highly of the dedication for that group from about April through July to produce all this data. But yeah, you can't take your experiment home, especially when it's a biosafety level 3 pathogen. And all these people are working in PAPRs, so air, that's not BL4. BL4 is the other part of our lab where you're working in a spacesuit. This is not a spacesuit condition. But when we do aerosol studies, yeah, you're working in a BL4 suite. You can't do that just anywhere.
Yeah, I think that, from watching the news, it's always, there's just a couple of people that doing the science and, then they find the right thing and then it gets produced. There are so many more, people in the background. And as you say, we know so little when there's a threat coming. And so when they say, well, we don't know if it's an aerosol, we don't know if that, somebody has to be there to test all that, to work behind the scene. So yeah, I think you and the team have been, the heroes in that. And when you talk about threat prevention, you are the guys that basically help us stay safe when there's threat, whether it's a natural threat or man-made threat. So that's all great, and we are all grateful for your work. I mean, it's absolutely fantastic, and you shared a little bit. Now, let me do one disclaimer. George, you were here as a friend, and so you expressed your personal opinions, not those of your institutions. But I think this was a great talk, and it gave us a lot of information about what you and your team have been doing, and certainly give us a glimpse of a more positive science future. Thank you so much.
Oh, thanks for the opportunity to represent the work, represent all the science. And as you say, we're all in the front lines in this one. So thanks, Thomas, for this opportunity.
Okay, wonderful. Thank you and goodbye.
Goodbye.